Well, all right, here is chapter 4. This chapter will be the end of module 1. You will have your module test. That should be coming up soon, so be prepared for that. Remember, you can use your books and notes, but you cannot use search engines. Don't type them into Google or Bing or whatever. Don't call your friends. There's no lifelines. Don't talk to people. Don't have a tutor help you with the test. You can have all that when you do these quizzes, when you do any other work, but you cannot have that with the test. The test has to be stuff that you've created yourself. So if you use audio notes, that's fine too. I'm not really sure how you'd be able to pull that off well, but you have to produce it yourself. So just remember that. I've caught several students cheating, and that's very, very disappointing. So store my faith in humanity and don't cheat. So cells. We're going to talk about different parts of the cells. We were first observed by Robert Hooke. He made a very basic microscope. And the first ones you were able to see were blood, sperm, and small organisms living in pond water. So here are the 5 different parts, the big ideas, your introduction. We're going to go to the nucleus and ribosomes, the endomembrane system, energy, and the cytoskeleton, cell surfaces. So we're going to go through them pretty quickly. Just be aware of that. So the introduction to the cell. And so first of all, in order to see it, you need a microscope. The most frequently used is kind of the light microscope, and that's the most when you see a microscope in the laboratory, and that's kind of what everybody sees as a microscope. So light passes through the specimen and then through the lenses and finally projected into our eyes. And specimens can be magnified up to 1,000 times actual size. So it's a pretty good microscope, and you can see a lot of different things with it. Now this is an important distinction. Magnification is how much that increases. Resolution is how clear the image is. And so you can go to these toy stores and buy a telescope, and it'll say 1 million magnification. And you think, wow, for $50, that's great. 1 million magnification, that's a lot. What they don't tell you is, yeah, something may be magnified 1 million times, but it's going to be so blurry you're not going to be able to know what's going on. What you also need is resolution, the ability to distinguish 2 close objects as separate. My physics lab professor worked for NASA with some different things, and he had top secret clearance, and I once asked him, I said, you know what, the satellites, the cameras on there, what kind of resolution they have? And he told me that if you were looking up and you had a name tag on your shirt, you could read your name tag, that if it had 2 quarters and a dime, and there was 6 inches of space between them, that they would be able to distinguish the difference. So that is just powerful, powerful resolution. Not necessarily magnification, but resolution. So they have limitations, obviously. Human eye, we have limits to our resolution, rods and cones, the number of rods and cones, right? The ability to distinguish between small structures. So light microscopes cannot provide details of a small cell. So we use other microscopes for that. And light microscopes, we can look at microorganisms, animal plant cells, some structures. But in the 1800s, when we did that, this led to cell theory. And so there's that scientific word again, theory. which states all living things are composed of cells, and all cells come from other cells. So if it's a theory, it's never been shown to be wrong, and is backed up by a massive amount of evidence. So if you can show some massive living thing that doesn't come from another cell and is not composed of another cell, then we may have to redo cell theory. So here some things we can see. With the unaided eye, we can see some squid neurons, chicken eggs, frog eggs. We could probably see a paramecium, but that's about as small as we can get. With the light microscope, we can look at that frog egg, and we can look at plant and animal cells, and even see some mitochondria. Well, with an electron microscope, we can see viruses, ribosomes, proteins, and we're getting to the point where we can actually now see atoms themselves, and that is massively incredible. And so here are some conversions. When we talk about size, kind of lets you know what's going on. The micrometers, 10 to the negative 3 nanometer. Or micrometers 10 to the negative 6 and nanometers 10 to the negative 9. Kind of give you a better understanding. If you look right here, nanometer, 10 nanometers, right? And so this gives you a little bit of scale for that. So in the 1950s, we started using the electron microscope to look at structures. Instead of light, we use beam of electrons. And so they can resolve structures as small as 2 nanometers. So we can see kind of 3D and magnify up to 100,000 times. Scanning microscope look at detailed architecture of cell surfaces. Transmission electron microscopes look at the details of the internal cell structure. And so you need different microscopes depending on what you want to look at. And so here's a paramecium. Everybody loves a paramecium. Look at all that cilia or hair, and look at that groove. And so you can kind of see the outside structure. And here, is this a eugleniid? It might still be a paramecium. I'll have to look at the book. But you start to look at the internal structure. It's kind of a slice if you slice through the structure. And this gives a little bit of a 3D look, but you can also see the internal structures going through that. So cell size. It's got to be big enough to hold your DNA, proteins, different things needed to survive. But it's got to be small enough that it would allow adequate exchange for the environment. So this movie, 1950s, The Blob, right? Got this huge blob that's supposedly the single cell structure that kind of just absorbs people. Well, the problem with that is the time it takes for, let's say, water to pass through one super really long cell would be huge, and so it wouldn't be able to react with its environment. You need things to diffuse quickly when you're dealing with cells. So they need to be really small to do that, but they need to be big enough to house DNA and other things. So it's a trade-off. You can't get too big, you can't be too small, and it creates an optimization between those two correlations. And so this is surface area to volume ratio, and this is very important. This is why skinny people generally get cold before larger people. They have a higher surface area as opposed to volume exposed to the cold, where larger people have more of a volume, and so it takes longer for them to get cold. And surface area to volume ratio, if you did your extra credit, is a huge part of just the biological world everywhere. And this trade-off is everywhere in nature when you understand what it is. So you may want to be able to calculate that surface area, the volume for the test, and maybe understand a bit about it for the test. So the cells, small size, they have a plasma membrane and forms that flexible boundary. And so you got those phospholipids, remember those from last chapter, hydrophilic heads, hydrophobic tails, right? And so that's what the cell works with. So and the membrane proteins are either attached to the membrane surface or embedded in that phosphobilipid layer. And so some proteins are channels, some are tunnels, some shield from ions and other hydrophilic molecules, and so others serve as pump and actively work to get certain things through. And so here is your graphic, and so you got that phospholipid bilayer, and you got your channel proteins, and so the hydrophobic region of the protein is going to be in the middle, hydrophilic is going to be towards the outside or inside of the cell. And so there's proteins embedded in this phospholipid layer that they allow things in and out. So bacteria and archaea are prokaryotic cells, and all other cells are eukaryotic. So you're going to need to know the difference between these cells and be able to identify the difference between these cells for the test. So prokaryotic, plasma membrane, one or more chromosomes and ribosomes. Eukaryotic cells have membrane-bound nucleus and a number of other organelles. So prokaryotes have a nucleoid but no true organelles. So a nucleoid is not membrane-bound, but that's the DNA of prokaryotic organisms. And so the surface prokaryotic cell, it may be surrounded by a chemically complex wall, which is one of the ways we use to identify it. Have a capsule, which allows things like anthrax to survive for a very, very long time in inhospitable environments. Have short projections to help attach or do cell communication, or have longer projections that help it move. All those things can be on the cell. And so here we are, the fimbrane, where, so those are the smaller projections, and you have ribosomes and the nucleoid and the bacterial chromosome on the left, and then you have flagella that helps it move. So you see it has a cell wall and a plasma membrane, and there's a capsule on there. Okay, so things like E. coli, and so eukaryotic cells, they're partitioned and they have different parts to them. They're not kind of just this cell with a whole bunch of stuff. They're kind of in the middle. So the nucleus and the ribosomes are involved in the genetic control, and then you have endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, peroxisomes, and they're all involved in manufacture, distribution, and breakdown. So think of a cell like a manufacturing plant, and all the different things that would go into a manufacturing plant. Mitochondria, chloroplasts are the energy, and then you have structural support. Okay, so the internal membranes of eukaryotic cells, that's what partitions it. That's what compartmentalizes it. And cellular metabolism are the different chemical activities that occur in a cell. And when you look at the difference between plant and animal cells, almost all of the organelles are similar. The few exceptions, so if I ask you to identify a plant or animal cell on the test, you should be able to do this. Lysosomes and centrales are not found in plant cells. Plants have a rigid cell wall, chloroplast, and a central vacuole. So plant cell walls are kind of square, rectangular, where animal cell walls are not. So here's your animal cell. Okay? So if you look, not centrale, lysosomes, those are not found in plant cells, but just about everything else is. Mitochondria is found in plant cells, and usually test questions like to pick that apart because a lot of people think mitochondria occurs in animal cells but not plant cells, and so that's usually the one they like to pick. They have nucleus, nuclear envelope, and ribosomes. Both of them do. So here's a plant cell. You can see it's much more rigid. It's got that rigid cell wall going on, and it's got holes called plasmodesma, which allow fluid exchange between cells. But you have that central vacuole and chloroplasts that are not found in animal cells. So there's part one of the introduction to the cell. Go ahead, do the questions, and move on to part two.